start screen sharing as well, okay? So my name is Eric. I will be your instructor for this semester for Bio 20. I'm new to COS. I taught over the summer. I'm a microbiologist. That's my forte. So I do work at a clinical lab in the hospital. I also have worked in food microbiology labs in the Central Valley. Recently graduated from Fresno State in 2018 with my master's in biology as well. So I'm local. I'm sorry, I'm going to mute somebody real quick because, yeah, background noise. So yeah, I'm new, but I'm here ready to teach Bio 20. This is exciting. I know it's online. I do apologize for that. We're not getting the normal face-to-face labs that we are accustomed to, but this will work. This will do. So for the first part of today's lecture, I'm going to kind of show you what to do with the labs, a little bit of the syllabus, and then we're going to go through a couple of the PowerPoints, hopefully a couple of the PowerPoints. So chapter 1 and chapter 2. But if we don't get through them today, no worries. We have Wednesday as well to get you the information, okay? So the assignments for this week are going to be due by Friday. I'm going to give you a little extra time. Typically, I allow 2 days for assignments. That's going to be the normal allotted time after the assignment is due. And these are going to be things that are pertinent to the lab. So you're going to go in and do your lab, and then you're going to do the questions, and usually you'll get 2 days to complete those. But for this week, since everyone's new, I'm going to allow to the end of the week to get everything finished. Nothing too serious. Most of the labs that I've seen with the Lab Connect are pretty simple, pretty self-explanatory, but we'll go through them right now and how to access the material. And then once the lab starts, I'll send a new link, and then I'll be walking you through the labs a bit, okay? So, let me see. Okay, Canvas up for your class. All right. So can everyone see my screen okay? Some yes. All right, and I have a poll that I got to use too. 1 second. So if you can see this poll, I just want to see if it works, because I'm going to be using this to actually ask questions. If you can answer that poll, I'm going to give you about a few minutes to let you ask the questions, or just a minute actually. Or 30 seconds, everyone's getting it done. And I think for those of you watching the recording, you're not going to be able to access this poll, but that's all right. All right, I have a couple more people that haven't answered, but I think I'm going to end it there. Everyone that answered pretty much said yes. Share results. You can see the results. Am I right to assume? Yep. Okay, no one's answering me. That's fine.
Still looking up. Sorry.
No, you're fine. No worries. I know it's early. This is an early class. All right. So I guess the majority of you, out of the people that attended, have used Zoom before. Good. So you should be familiar with this. What's going to happen every morning is I'm going to send you a link directly to your email. And you're just going to click on the link, and you're going to access the live lectures. So whether one of you shows up, whether 10 of you show up, 20 of you show up, I'm going to always have the live lectures Mondays, Tuesdays, Wednesdays, and Thursdays, okay? Let's say if you want to attend the live lecture and you're in a Monday section and you want to attend the Tuesday section, you can go ahead and, oops, someone's having trouble logging in. You can go ahead and message me, and I can send you the Zoom link for Tuesdays, okay? So that's for individuals that just either want to attend the lecture a second time if they didn't get something, or if you just missed that Monday lecture and you would like to have a live face-to-face lecture with me on Tuesday or Thursday, that's more than fine. Okay. If the same thing with the labs, if I'm going to give a small little lab lecture that you missed or that you wanted to ask a couple of questions in, please stop by that lab on a Tuesday or Thursday. So I know your sections meet Monday, Wednesday, but again, if you want to meet it in a different section time, that's fine. Okay. Just let me know. Start screen sharing. All right, so here's Canvas. And everything you need to access for the most part on Canvas is going to be in the modules, all right? And this will be your home. And this will have the lectures for today as well as the syllabus because we just started the class. So you can access the syllabus here. You can access the 2 PowerPoints that I'm going to be lecturing on in a few minutes, okay? Another thing you do need to know about Canvas is that we have a couple of different links or a couple of different software tools that we use. These are the software tools that you don't see yet because I haven't uploaded any quizzes or assignments, okay? And every now and then I'm going to upload an assignment onto Canvas and it'll just be the assignment that's assigned on the schedule. Or I will upload a quiz that will be accessed by you through Canvas. Okay, there are going to be assignments that are due through McGraw-Hill Connect, and that's going to be your lab resource. And that's a separate thing on its own. You're going to get separate points for that, but you have that weekly quiz that's due. And don't worry, this week's quiz is pretty simple. I just want to see if everyone can access and use it in Zoom, okay, and perform the quiz. All right. So I'm going to show you how to access your McGraw-Hill. This is linked to your Canvas shell. And what you're going to want to do is click on Go to My Connect section, okay? And what's going to happen here is going to prompt you to log in or buy or purchase a Connect code, and you don't need to purchase a code, okay? I can't say that, stress that enough. We have the codes provided by the campus. I will be providing you with a code once I receive them. Since I don't have them yet, you're going to need to enroll into a temporary or like a trial or test run for the online software, okay? And that'll last for however long it is. I should have the codes before that trial runs out, most definitely, okay? Hopefully by this week I'll have the codes and I can send you out your individual codes and you can have access for the full semester, okay? So if you click on that link, it'll take you to the Connect Biology, okay? And this is something you should be able to access once we start lab today, okay? So when I send out that second link, that second Zoom link for lab, which is I believe at 9:45, I'm gonna start here, okay? So everyone should be able to go to my Connect and then access this. So you will not see this page initially. You're going to have to enroll or for that temporary access code, and then it'll bring you to this page. Okay, so just kind of keep that in mind. But yeah, so again, you're going to come here, McGraw-Hill, click to go to my section, and then again it'll prompt you to sign up for that temporary access code, okay? And that is it for showing you around Canvas for now, okay? And if any of you have any questions, go ahead and email me. But yeah, we'll go through the syllabus and talk about office hours and things like that right now. Let me see. All right. So please let me know if you have any typos. I had to work on this syllabus quite quickly before the start of the semester. So go ahead and let me know if I screwed up or anything. It would be probably the easiest way for me to amend the syllabus. Okay, so this is me, Eric Arteaga. I have my email here, so go ahead and email me whenever. And I'll say that with kind of, yeah, you can email me whenever. I usually get back to you within 24 hours, okay, even on the weekends. So again, I do work at a clinical lab at Cuya Delta. So I'm usually up at weird hours. I think I sent out the link for the Zoom lecture at a late hour early this morning, I should say. So I'm usually up, okay, and I usually respond to emails quickly. So please email me, okay? Office hours are going to be Tuesdays, 11:30 to 1:30 p.m. I also have an office hour on Thursdays for my micro sections. So if you would like to attend that, if you're unable to make Tuesdays, you can request for that Zoom link for that Thursday time as well. Just to say that the micro students will have preference at those office hour times. But if you do want to chime in and get some help with bio, then or intro bio, then I can help you there. Okay. So again, this is Bio 20. You can read this on your own. I know this is a lot of the course outcomes, and I'll be talking about this throughout the semester anyways. I think we're going to go over the points. So required materials, Campbell Essential Biology with Physiology, the 6th edition. You can look up the book through this ISBN number as well. If anyone is having difficulties purchasing the textbook, please email me. There are a few copies that are available for free to rent, but I was told that this is for individuals that have the inability to purchase the textbook. Okay, so please email me directly, and we can get you a textbook if you're unable to purchase the e-text or rent the textbook. Okay, I do recommend the e-text. I'm using the e-text to use for the lectures and for the test materials and for the quizzes. So yeah, you do need the textbook. I recommend the e-text. It's $40. Like it's relatively inexpensive compared to the actual hard copy. Okay, the hard copy is pricey. It's like over $100. Okay. Online access to McGraw-Hill virtual lab. I will provide you with this again when you go and you sign up online and you don't have the access code right now. Just sign up for the temporary or the a trial run using that software. Okay, and you do need a computer with internet access. And here's the first typo. Please see me or please email me if you are unable to or you don't have the proper equipment or you don't have a computer. Okay. They are loaning out Chromebooks with webcams and various other hardware for through the campus at COS. So please let me know if you're unable to access or you do not have access to a computer. We can get you one. Okay. So please let me know. So attendance. I know this looks scary, but this is written for face-to-face labs. But I will drop you if you are not completing the work on Canvas or through that virtual lab. If you're having issues accessing that lab or you're having issues with Canvas, please let me know. Please notify me. I can help you out. I can help you understand. We can have a Zoom meeting and I can walk you through the steps of how to use your Canvas shell. Okay. And it can definitely be a one-on-one thing if you don't want to have, or you don't want to chime in during office hours and everyone's there. That's fine. But please let me know if you're having difficulties. Okay. I do not take roll through the Zoom. Again, I take roll through Canvas. So the moment you complete your assignment on time is the moment I check you in as you're here in class. Okay. Again, this is an asynchronous and synchronous course. So I do have a lot of FaceTime with you if you choose so. If you don't, that's totally fine as well. You can continue the course without ever having a conversation with me directly. Okay. You can watch the videos, complete the assignments, and get through the course that way. All right. I don't recommend it. I honestly recommend that you show up and attend the live lectures and the live labs. And if you have any questions, you can ask me directly then. Okay. So again, I do recommend attending the live lectures and the live labs. And it kind of holds you to that standard of being up, listening to the material, asking questions if you have them. Okay. So here's a point breakdown. We have 10 quizzes. So you should have one a week for till the end of the semester. There's an extra credit quiz at the end. That's quiz 11, and you'll see that on the schedule right now. So you do have lab assignments, 10 points each. So you'll see that the lab is worth about 5 points, and the questions are worth 5 points as well. So half the lab you earn credit just by completing the online simulation, and then half the points are also awarded through the questions. Okay, and if anyone is having issues answering a couple of the questions, just let me know. I can walk you through. I can walk you through the questions as well. Okay, so they're not made. Those questions aren't there to quiz you, or they're not there to be too terribly difficult. I just want to know that you understand the material and that you understand the lab. Okay, so again, 5 points for the lab, and then 5 points for those questions. Okay. Exams. So there's 100 points each, and there's going to be 5 exams. Okay. There's quite a bit of exams, I'd say, for a college level course, but again, there's a lot of material that you're going to be provided throughout this semester, and it's very broad biology. Okay, so there's quite a bit of information. It's not too specific in terms of what biology gets down to in the upper division classes, but it is a lot of material, and it might be material that you've never experienced before. Okay, so please, if you need any help, if you have any questions about any section, let me know. Talk to me. I can help you with tutoring, or I can provide you with that tutoring service if you need extra help. And we do also have a tutoring center at COS, and it's referenced here. Okay, so you can access that tutoring center. Oops. I guess it's right here. Okay, all this tutoring center information is here. Okay. All right. You do have a midterm, and it is cumulative, and it's only worth 100 points. So it's just an extra test. Okay. Science topic and paper. So this is kind of the big one that you won't get a lot of information for right now. I'll kind of talk to you maybe a third way through the semester. I'll kind of talk about this a bit, but you will have a paper that's due right around 1,000 words to 1,500 words. So small paper with a presentation. Okay, so I will be meeting with you individually, and you will be giving me a 10-minute spiel about your science topic of your choice. Okay, so it's usually a science topic that's pertinent to your field. So if you're in business, you can talk about some sort of business or biological topic that deals with business. And again, you're going to write a paper about it, 1,000 to 1,500 words, and then you're gonna give me a little a presentation. You can do an oral presentation. You can have a PowerPoint. It's really what you want to do. Okay, PowerPoints usually help, I feel, with individuals that are kind of timid when it comes to talking or presenting. PowerPoints can keep you on track, kind of help you through that. So I definitely recommend a PowerPoint for this. And again, it should be a 10 to 15-minute spiel, and I'll be asking you a couple of questions about your topic. Okay, and your topic does need to be approved by me. All right. Here's a grade breakdown. I'm usually pretty firm about grades. I usually don't curve unless the class is doing really bad. But for this class, I doubt that we're going to have too many issues. Okay. All right. So I think that's pretty much it. So scheduling. So next week is your first quiz. So that's your first opportunity to really be tested on, but you do have point opportunities every single week. Okay. So every lab is worth, every week's labs are worth 20 points. Okay. So even if I have one lab that's scheduled, you'll have multiple labs that will be worth 20 points. All right. And again, that lab section is worth 5 points for each lab completed. And then those questions are worth another 5 points typically. So you'll have 2 labs that deal with chemical composition. Okay. And every other week you should have a quiz, if not every week. So again, the only time you don't have a quiz is when you have an exam. So week 4 is your first exam, and you're dealing with chapters 1 through 6. Okay. So again, there's going to be a lot of exams in this semester, but again, I think this is better in terms of helping you study and helping you navigate the material. Again, the final is cumulative, so you will have to recall information back from chapter 1. Okay, but luckily the information builds on itself. So yeah, if you have any questions, please let me know. Other than that, I guess I'll start. I will be starting the lecture. Questions, concerns? No? Okay. Alrighty. So we end at 9. I think 9:15. We'll go to 9:15. Sorry. All right. So chapter 1. All right. So what is biology? So biology is the scientific study of life. Okay. So biological studies can deal with large mammals to microscopic organisms such as bacteria. I will be talking about bacteria a lot in this class because I love bacteria. That is my specialty. I am a microbiologist. So when we talk about ecology and things like that, I will be referencing bacteria more than, let's say, ecological systems found in the rainforest, okay? Because that's what I know about, all right? So biological studies can also deal with life processes such as genetics and proteomics. And I know proteomics is a scary word. We'll talk about it much later, but it's talking about the functioning of proteins, okay? So biology can deal with individual cells, individual chemical processes, all the way to understanding how lions are hunting in the savannah, okay? So a very broad field, and it deals with a lot, okay? So it's important to know. And again, it is the scientific study of life. All right, so the scientific method, this is going to be the method that we use to actually test or identify problems in science, okay? And this is a way to organize our thought process and a way to actually see if something's interesting and actually test it and see if there's something we can learn from that biology, okay? So science, any method of learning about the natural world that follows the scientific method, okay? So science, remember, we have very specific rules on how to measure and how to identify problems. And these problems can then be looked at in a very special way in order to determine if they are interesting or not. And I say interesting in terms of whether or not they're going to provide some sort of understanding of the natural world, okay? And which is very important, right? We still know very little about the human body. We still know very little about various ecological systems. And you hear this often, right? The space, or we've been to space more than we've been to the bottom of the ocean, right? There's a lot of areas on Earth that we have not discovered. There's a lot of biological systems that we have not looked at specifically. And the scientific method is a way that we can experiment and actually understand those systems, okay? So the scientific method, method that is used to scientifically test a prediction. You must make preliminary observations and collect data before you are able to formulate a testable prediction, okay? And what this means is you have to look at a biological model or an issue or a problem, and really look at the data and really look at that problem in a scientific mindset, okay? And what that means is you need to collect preliminary data or observations, which means you need to look at that, whatever that problem is, identify it, and then look at all the different parameters that deal with that situation, okay? And this is very important, right? You wanna make sure that you're doing the proper steps in order to make a good scientific study or good scientific experiment, okay? Hypothesis. This is a tentative explanation that a scientist proposes for a scientific phenomenon that has been observed, okay? And what I know people have talked about hypothesis as being an educated guess. This is more of an educated statement about what you have observed, okay? We'll kind of talk about that a little bit later, and what a hypothesis really is in terms of what you are saying about your observations or what you're saying about your research or your experiment, okay? It's going to be a statement that you use to explain what you think may or may not happen, but it's just a belief, right? It's just initial start of some sort of understanding of that issue. All right, an experiment. A scientific test often carried out under controlled conditions. that often involves changing just one variable, okay? So in an experiment, again, changing one variable is kind of the standard, the gold standard of what you uh, and what you're conducting your experiment on. Um, you want to make sure that you're only manipulating one thing because that's the one thing that you're testing for, okay? If you start changing multiple facets of that experiment, you're going to have multiple different outcomes that you might not explain, right? I think the book gives an example of cookies, right? You want to make the best batch of cookies you could possibly make. And for that experiment, they changed the flour recipe, okay? So you don't want to be changing the flour and the sugar and the chocolate chips, right? You want to be changing one facet to make sure that you know you're making the best possible cookie, right? So let's say if you do change the sugar content, or you do change the flour and you change the chocolate chips, you're never going to know what parameter is actually making the best cookie, okay? So you always change, or not always, but you typically would want to change one parameter or one variable within that experiment to actually see whether or not something is working or not working in a certain way that you think it is, okay? All right, so the scientific method kind of runs in this flow chart. You have exploration or understanding or data collection here. You have your hypothesis. So in terms of this, you're wondering why the remote isn't working. So the remote's batteries are dead. That's your hypothesis, right? The remote isn't working because the batteries aren't dead, but there's a lot, or the batteries are dead. So there's a lot of different parameters that also can deal with the remote as well, okay? The remote could be broken. You might not be pushing hard enough on the buttons, right? The remote is the wrong remote for that specific television, okay? But for your hypothesis here, you're using the batteries as the main topic, okay? Right? So testing is very important also, again, to forming that hypothesis to make sure to understand that either the batteries or it's the buttons or it's the remote being broken, and you can draw conclusions from that testing, right? You can change the batteries, you could turn on and off the TV, you can do something that changes the parameters, but again, you want to make sure that you test one thing at a time, okay? So here's a prediction. If I replace the batteries, the remote will work. Okay, and that goes off of your initial hypothesis. You're testing something here, which is changing the batteries. Okay, experiment. I replace the batteries with new ones. So experiment supports the hypothesis. The remote does work after you change the batteries. Okay, so this is kind of going down that flow chart. I know this is kind of a boring explanation. It deals with the remote, not too important. But again, this kind of shows the thought process when it comes to scientific thinking, right? You're looking at one specific problem and you want to change one specific parameter to see if it's going to work in your favor or against your favor. Let's say those batteries, once you change the batteries, the remote still didn't work. Well, now you have more testing to complete. Now you have to look at other different parameters that are going to make that remote actually work, okay? All right. So I talked about variable earlier. So variable is a factor that is changed in the experiment. We typically usually change one factor, okay? We also have something called a control group. The controlled group is the group which everything is compared to, so nothing is changed. So many times we think of a control group as a placebo group, especially when we talk about drug trials, right? I believe when they did the testing for the coronavirus vaccine, they injected people with a placebo vaccine, right? So that's a vaccine that does not contain anything in it. It's just going to be like saline or something that will be injected and nothing will happen to that individual, okay? And they want to compare that to someone that was injected with the actual vaccine, okay? So the control group is where you do not have any of the experimentation completed on, all right? Let's say if you were testing the cookie, what made the best cookie, right? You're changing the flour. You're going to stick to the original recipe for the control group. Nothing will change from that group, okay? The experimental group, you'll have the flour change, okay? So segueing onto the experimental group, this is a treatment group, right? This is a group receiving the vaccine. This is the batch of cookies that's having the flour changed. So this group that receives a changed variable or treatment, okay? Another thing to note is that, as I mentioned before, especially with the batteries in the remote, if that didn't work, you would continue to make different observations or different tests or conduct different experiments. this is an ongoing process. Okay. So there's a lot of different factors that come into what you're testing and how you're testing that specific variable or that specific experiment. But after failed hypotheses or let's say it segues onto something else, you can continue making different observations and continue with that experiment. Okay, you can test new hypotheses. You could perform new tests. You can further strengthen your knowledge on that specific topic by changing or adding different experiments or variables. Okay, so it's important to know that this is a very fluid process. Typically, scientific research leads, or scientific discovery leads on to new scientific research. Okay, one of the biggest examples I give in my microbiology class is HIV/AIDS. Okay, HIV was the first virus to ever be discovered to have something called reverse transcriptase. Okay, and reverse transcriptase is something that can convert RNA into DNA, which has never been discovered before. But terrible as HIV was in the 90s, it also provided us with a lot of new technologies, and especially with identifying different types of viruses. Okay, so that one enzyme discovered in that virus gave us a lot of different molecular assays that we're able to identify viruses that we were never able to identify before, never thought we would be able to identify. Okay, so again, discoveries will lead on to new research and new experimentation, and hopefully something that the world can utilize. Okay, and with HIV was discovered terrible, but then we had that protein that was identified from HIV, and now we can utilize that in molecular testing. So again, very cool, I guess. So quality control. So it's important to have your research peer reviewed or your experimentation peer reviewed. You want to make sure that you're doing good research. Okay, so this is very much a part of science, right? And many of you probably will never go down this route in terms of performing research. I think there's very few or very finite people that go down research. I only went through research just in school. So my research experience is very limited in terms of doing this on an everyday basis. But peer review is very, very important. Okay, the evaluation of scientific work by impartial, often anonymous experts in the same field. Peer review is considered a good means of recognizing valid scientific experiments. Okay, so what does this mean? So it essentially means that your research or your experimentation must be checked. Okay, we cannot make unsubstantiated claims. Sorry. This tea will cure your stomach pains. This salt will get rid of your acne, and so on. Okay, so claims must show proof, right? And this is very important, especially when we get into pseudoscience, or especially when we get into claims that are made by, let's say, that random ad that you see on your social media account, right? This powder will give you immense amount of energy. Okay, you drink this 5 times a day, and you'll lose 50 pounds, or you'll be able to run twice as long. Okay, well, how? Right? We want to make sure that we're looking at the science, and we're looking at how that product or how those claims are being tested. Okay, so and science is reviewed this way, right? Anytime there's a drug trial, anytime that we or scientists or physicians release some sort of new drug, it goes through a process of checks and balances, and checks the validity of that drug, whether or not it's useful, whether the harm or the gain outrisks the harm, right? So it's important that we note that peer review is a big part of science. Okay, we want to make sure that people that are doing the experiments are doing them truthfully, and they actually do work. Okay, one of the big things that we see in the laboratory is, especially when I was doing my thesis, was that if I did an experiment and it worked, I had my my partner or my PI or personal instructor actually complete the test next to me as well. And that was to make sure that I wasn't doing something different from anybody else, that everything that I was documenting was the truth, and that there was something that I wasn't missing, right? Because if he or she was unable to perform that test, then I'm doing something wrong, or I'm doing something different that's giving me a different result. Okay. So the results must be repeatable, and you must show proof that they're actually working. Okay. All right. So again, this is referring to what I mentioned earlier as repeatable. Controlled experiments should be repeatable, which means someone else can repeat the experiment with their own reagents and instruments. Okay. So typically, what you see with very highly regarded science in most cases, you see that someone will publish a paper and another lab will contest it, or another lab, even in another side of the world, will look at that research, and then they would want to repeat that experiment. Okay. And they want to make sure those claims are true, especially if it's groundbreaking, especially if it's something that's changing the field. Other people will want, or other scientists will want to make sure that that experiment is repeatable, just to make sure that that lab essentially isn't full of it, right? You want to make sure that they're doing good science, and that that lab is performing everything correctly. Okay. All right. So this is kind of a nice little diagram, kind of showing you that testing is key to communication outcomes and exploration. So exploration, again, you're observing something, asking questions, seeking information. And this can be anything from a new field of study. This is, again, can be referring to uh, uh, exploring new environments. Again, biology is very broad, and again, it encompasses a lot of different fields. Okay. So testing is kind of at the center of all of this. Again, gathering data, interpreting data, drawing conclusions, messing with that data. Statistics is a huge part of science as well. But again, you won't really have to deal with that in this class. Again, I just want you to be able to identify the different components to science, and different components to the scientific method, and how we utilize these different steps to kind of come to our conclusion and come to our theories and different hypotheses. Okay. So segueing on to theory, a widely accepted explanation idea that is broader in scope than a hypothesis generates new hypotheses and is supported by a large body of evidence. Okay. So one theory you can think of or talk about is, well, I think of microbiology, but I can't think of one that's probably not dealing with microbiology. But the germ theory, let's say the germ theory, that's if you are successively less exposed to bacteria, you're going to develop a hyperreactive immune system, right? So people back, 2,000 years ago were exposed to more types of microorganisms than now. So their immune systems are a little more different compared to ours now, right? Where we clean everything, so we have more autoimmune diseases and things like that. Okay, so again, referring to that theory, right? There's evidence that's backing that up from different claims and different experiments. Okay, so fact is a piece of information that is correct based on all current information. A fact is not subject to opinion and can be independently verified. Okay, so a fact would be gravity exists, right? That would be something, that would be a fact. It's not something that I can say or that anyone can say that is false. Your opinion is not a fact. So you say, your opinion on the way something looks, like this flower is beautiful, not a fact, right? We cannot substantiate that. There's no way to test that. A fact will have to be something that we can actually test. We've tested gravity and how it works for many hundreds of years now. So now we know that it exists, and it's a fact. Okay. All right. So controlled experiments. So a good experiment typically only changes one variable, as I mentioned before. So you do need to know this. Okay. So we have blind experiments. So this is an experiment where information is withheld from the participants. You have a single blind experiment. Info is withheld from test subjects. Okay. So information is held from test subject. This is typically what we see in drug trials. Okay. Where the researcher knows of the experiment and knows what they're giving their patients, right? They need to know in order to monitor serum antibody levels, or check to see how that patient is doing. But again, those individuals, those patients, or those people that are receiving the placebo or the true vaccine don't know. Okay, you don't want them to know. All right, you want to make sure that whatever they're receiving that treatment um, is going to be not have like a bias reaction from that individual receiving those treatments. Okay, so a double-blind experiment is one where info or the information is withheld from the test subjects and the researchers. So typically, you see this with well, I've seen it with psychological studies where you're administering some sort of treatment or administering some sort of test, uh, and the experimenter doesn't know the age range or what they're providing or what type of treatment they're giving the individuals receiving that treatment. Okay, so again, you want to make sure that you're not providing a bias towards the test group by the researcher. Okay, I know that sounds kind of crazy, but it's true. All right. If I was performing an experiment and I really wanted it to work, I'm more inclined to actually do something to change the parameters in order for that experiment to work. Okay, so it's important to utilize this strategy sometimes when we're looking at a very specific project or a very important project, and you don't want that researcher's biases to kind of spill over. Okay, again, that's important in psychological studies because again, we are born and we grew up in a different background. So all of us have inherent biases that we can transfer onto that experiment, especially when working with people and experimentation with people. Okay. So again, single blind information is withheld from test subjects. Double blind information is withheld from test subjects and the researchers providing those treatments. Okay. So I know I referenced a placebo earlier. So a placebo, harmless but ineffective procedure or treatment that is given purely for psychological reasons or to act as a control in a blind experiment. Okay. Again, this is going to be something. Best reference or best example I can give you is a vaccine trial where there's actually a true vaccine which has the antigen or that actually vaccine that's being made, or you have like salt water that's being injected into the individuals. Okay, the salt water is harmless, or the saline is harmless, and then that actual vaccine is being injected into those other individuals. And again, you want to compare the placebo group to the individuals that are receiving that treatment, again, as a control. All right, there's a nice little chart that kind of shows you single blind versus double blind. So not blind, both test subject and researchers know about the treatment and what they're receiving. We have a single blind, which is the test subjects don't know anything, whereas the researchers do know. And then you have a double blind, right? So both researchers and test subjects don't know what is going on, okay? And remember, for the double blind, it's to remove that bias from that researcher, okay? So again, if you really want an experiment to work, you might be coercing your test subjects into picking the right tests, okay? And this could be, again, mainly used in psychological studies where the researcher is administering either a verbal test or like an eye test or something that something where the human biases can get in the way. Okay. All right. So features of science versus pseudoscience. Again, I mentioned pseudoscience before, and again, this is mainly referencing where the best example I can give you is when you go online, you see those products that are making these unsubstantiated claims where drink this powder, drink this tea, and you'll get taller, you'll get way more energy, you'll shed 20 pounds in a month, which is very unhealthy. You shouldn't do that. But again, those unsubstantiated claims, again, that is referring to pseudoscience, where these claims are not tested. These claims are not accepted by the scientific community, whereas features of science are going to be the adherence to an established and well-recognized scientific method. Again, what I outlined previously. What I gave you previously is a very broad kind of scheme of how science is done, okay? We do have repeatable results, so other people can perform your tests and they can get the same outcome. Whether that lab is in New York or in LA or in the Central Valley, they can do that test no matter where they're at, and they're going to get the exact same results every time. So testable claims that can be disproven. So this means that you have all the data, you have all the background, and you also have all your I guess paperwork in a row to show what you've done. Okay, and once you've shown what you've done, you then have the ability to disseminate your research or pass it around in the scientific community for other people to test and to make sure that you're doing the proper work. Okay, if you're not doing the proper work, you'll get called out, which is good. You want that. If someone makes unsubstantiated claims, you want to make sure that they get outed or that everyone knows that that's not true. Okay. Open to outside review. Again, this is referring to peer review. They want to, and this is mainly referring to writing up your experiment and ensuring that that your research is solid, that you're doing the proper steps, that your results are not drawing too much claim out of them. Okay, that you're not your conclusions aren't expanded too much on what you've done your research on, right? So, and this is mainly referring to people utilizing the research to explain something much more broad and much more open. So the example I like to give here is when slushy sales go up, violent crimes also go up, right? So you're equating the slushy claim to violent crimes in the city, okay? Which is kind of that's crazy, right? The higher the Slurpee or slushy sales are, people are more violent. That's kind of an unsubstantiated claim that requires a review, right? You can make those correlations, but again, that doesn't really make sense. What makes more sense is that slushy sales go up because of increased temperature. Violent crimes go up because of increased temperature. People are hot, more upset. They have, they don't have AC, let's say, or people become more violent or aggravated when it's extremely hot outside. Okay, but also slushy sales go up. So that might be a better way to explain that situation, right? You can't equate slushy to violence. It just doesn't make sense, okay? And the review process will kind of tease things out like that, right? If I make a discovery in a field and I make a bunch of claims about it curing cancer or it saving lives or having some sort of implications of medicine, I need to have the proper documentation, the proper experimentation to prove that, okay? So again, multiple lines of evidence. So you test multiple facets of that project or that research, okay? All right, so features of pseudoscience. So it does not adhere to generally accepted process of sciences, results that cannot be duplicated by others. Again, you can't send this to another lab and have them perform the same test and then get the same results, right? It's going to be